So I guess we'll start with the very first lecture, and for many of you this is really just a review of information that you already have, but not knowing who is going to be taking the class, we prepared some slides, and we're going to talk about some existing technology. Many of you already said you're very familiar with existing technology. Some of you are fairly new to this. We're going to talk about detection of illicit substances, and by this I mean drugs and explosives is what we're referring to here. We're going to talk about how the current technology detects particles, residues, and we'll call this trace. And that's why the TEDs, trace explosive detectors, are referred to. Trace is of minute quantities, not visible with the naked eye. So we're going to try to detect those. We're also going to detect the vapors, and Brian indicated he's interested in vapor detection, and many people are now, because if you think about it, you know, this is exactly what the canines do. They're detecting the vapors, the volatiles, the odor signatures. We're going to be talking about these terms interchangeably. And if you think about it, canines detect odors in a room. So you'll have a demonstration of a drug dog in a little while at 11 o'clock this morning, and you'll see right next door here how the handler can place the illicit substance one place in the room, and the dog can go and trace and go and follow the odor to where the location of the device is. There's a professor who studies canine olfaction who wrote this interesting statement about dogs can detect the present, the future, and the past, not just the current state of the presence of volatiles, but if you think about it, if you've got wind blowing downwind here, and the canine is here, the explosives is downwind. That's the future. So the explosive is going to be in the future as the explosive as the canine is approaching the explosive. It's also the past. Somebody has deposited some explosive in the recent past, and as you are walking the dog by, they are detecting the past. So that's kind of an interesting way of looking at it. We are going to talk about biological detectors, including this, the demonstration. We're not going to get into a lot of the theory and the biology of canine olfaction, but we will talk a little bit about the theory and the chemistry and the instrumental analysis of instrumental detectors. We're not going to get bogged down, but I'd like to give you a little bit of a background there. So I don't have to tell this group how important it is to detect these illicit substances, and I'm talking about explosives and drugs. We have many examples of how important it is, how big of a threat it is. I was listening to a talk recently at a conference in which 100% of all vehicles in London at the upcoming Olympics will be searched. 100%, one way or another. Now, there is no perfect tool, as you know, and you have to look at it as a series of tools in your toolbox to give you the best chance to detect. In these cases, they were not detected. We have other examples. Christmas Day bomber, not detected. Got even actually to the point of trying to detonate the device on the plane. Not detected, even with all the great tools that we have already. So think about it as having a series of tools in your toolbox and then taking them out and using them. And it's up to you to identify what's the best situation to take out that tool. So I'm not going to talk about the problem. We know we have a problem. There are threats, and we're trying to detect them. And so we have a list of types of explosives. There's a variety of explosives, so we can't focus on one. We don't know what the threat will be. Let's go from low explosives to high explosives. We have commercial explosives. These are difficult to get a hold of, and even more difficult are the military explosives. And so we have decided in our research to focus on explosives that are relatively easy to get a hold of, low explosives. And low explosives deflagrate, so they have a speed of reaction that is less than 1,000 meters per second. And you can buy these, you know, fairly easily. Black powder substitute, smokeless powders. These can be used to form a bomb. And so we have decided to focus on these. These other high explosives, they detonate with a much faster speed of reaction. And normally, the low explosives require an oxidizer. High explosives contain the oxygen within the compound itself. So the fuel and the oxygen are contained within the same compound. TNT, for example, has a source of oxygen and a source of carbon and fuel, so does not require oxygen. It's self-contained. So we have these commercial explosives, and people who do mining, quarry work can get access to some of these, not easily because they are controlled, and especially the detonators are very controlled. And then we have military explosives are also very controlled. But the black powders, the smokeless powders, are explosives. They're easy to get to. I mean, we purchased some for this research, and so we figured let's focus on those. But a lot of the stuff that we're going to talk about is applicable to both. When we have done a lot of research in conjunction with colleagues at other locations where high explosives can be handled, like the Transportation Security Laboratory and other locations where we can go and send our students and do analyses. So a lot of the stuff that we'll talk about pertains to high explosives and military explosives, commercial explosives, and we'll talk about that. But all the exercises we're going to do this week are the low explosives. We're going to talk about detecting smokeless powders. Okay. Now, detection, bulk systems, bulk analysis. So we're going to detect the main mass of explosives that are hidden inside containers. So we have different technologies already available for this. X-ray technology, neutron activation, millimeter wave. So these are to detect a bulk amount. And then we have trace. So we'll make a differentiation between bulk and trace. And then within trace, there are two subgroups. One is the laboratory detectors. And so if you go to a forensic laboratory, you will see gas chromatography, mass spectrometry, you'll see capillary electrophoresis, and you'll see, mass spectrometry systems. So these are unambiguous detection in a very controlled setting where these could take a lot of time. The GCMS analysis, for example, is on the order of 45 minutes just for the analysis. The preparation of the sample takes longer. Running a blank before you do the analysis, which is also very important, and you're going to have to do this when we do the exercises. You want to make sure that the entire system is not giving you a false positive. So we do a blank, then we do the sampling, then we do the analysis. So this takes time. You don't have this kind of time in the field. So we have field detectors. Somebody mentioned FIDO. There's a picture of FIDO here on the right-hand side. Ion mobility spectrometry is a very nice tool for detecting illicit substances. They're very, very fast and can be very portable, can be taken to the field. But by far, the number one method for detecting illicit substances in the field are canines right now. That is the most effective, efficient method, okay? So many agencies, federal government, state, local, utilize canines for drug, explosives, and other threats. So if you want to take a look at the limits of detection, how low can you detect something? I have a graph here. This is the phyto system, fluorescent polymers. So there's a reaction that happens, that a chemical reaction that is triggered with the presence of, say, TNT on these polymers, and it's extremely sensitive. So if you look at parts per trillion in terms of the presence of explosives, we have the phyto system beating out everybody else. But this is not particularly, well, it's very selective, okay? So really, it works really well with TNT, and they are developing other threat detection for the phyto. And Tom, maybe you can tell us later, you know, what your experience with the phyto has been. IMS.
I'm sorry.
Yeah.
To interrupt, Odor.
Go ahead.
I'll tell you now.
Go ahead, please.
And again, maybe the technologies have increased over the last, it's probably been 3 years since we actually attempted or played with it. We actually worked with them for a week. The problem that we had with the phyto is you had to be right on top of the explosive to really detect anything. So if I was a foot away from it, it wasn't detecting anything. So I had to stick my hand in a box, and that just wasn't feasible for our jobs.
Yep.
Or for first responders. Now I know that they are using it in theater, so maybe their technologies have changed, or maybe they've gotten better. I'm not sure.
Mm-hmm. Yep. Thank you.
Is that still the case?
You know, I don't have any experience with the phyto myself. I do know that they are constantly improving these technologies, so I'm not sure, you know, how much better it is. But you know, like I said, there's no perfect tool, and it just happens to be, in terms of sensitivity, we have to note that it's very, very sensitive. Now, if you're going to use it as a vapor detector, you have to make sure that you're targeting something that has an available vapor. So not only is the technique itself sensitive, but you have to get the sample to the instrument. And so that's important, so keep that in mind. IMS, in terms of sensitivity, is the next best thing. So in terms of limit of detection as an instrument, it's the next best thing. Again, the key is to get the sample into the instrument. And when you have particles and you swab the right area and you get those particles onto the instrument, it works great. It's a wonderful tool. Mass spectrometry that's a laboratory system, works really well. And then canines, the sensitivity is dependent on the way you train the canines. We can train canines to go extremely low, that's just a function of, in terms of the sensitivity, is how do you train them? Okay, we can get into more of that later. The effects of the properties of explosives on detection. Explosives have these properties, we'll call them electronegativity. That's the ability of the affinity for electrons for a substance. Fluorine, for example, has the highest affinity for electrons on the periodic table. Very negative element. Explosives in general have very high electronegativity. Adsorptivity, thermal stability, so explosives are not very stable thermally. They decompose because we want them to. As a designer of an explosive, you want the compound to break up and detonate really quickly. And on the other side is you want a compound to be fairly stable so it doesn't go off unexpectedly. Frangibility is the way that the material breaks up. So cookie dough is not very frangible. It doesn't break up, but a cookie does. A cooked cookie will break up into little pieces, and that's one of the properties of explosives. So they'll break up and fragment. And then this other thing, vapor pressure. This is important for us because we can use this as a detection tool. So we can use the vapor pressure characteristics of an explosive. Many explosives, like RDX, do not have a very high vapor pressure. That means they're not available to be smelled. So they're not available to be detected. So we have to look for another property or another associated odor to RDX. And that's exactly what we're doing in many cases. So here are plastic explosives. We have RDX, PETN, and then mixtures. What we're going to do is we're going to look at the plasticizers. So these compounds right here, we're going to use at the binders, we're going to use at other compounds, and military explosives should have a tagant that is very volatile, DMMB. We can target this tagant. Now, if you're a bad guy, especially like a bad guy with a lot of resources, you can get a hold of plastic explosives that do not have the tagant. So we can't hang our hat on looking for the tagant in every case. So we'll look for the tagant, but we'll also look for these other compounds. Now, most ETDs are going to look for just RDX. So far today, if you expose some unknown compound to your ETD, it'll look for RDX. If it's there, it'll alert to RDX. But currently today, we're not looking for these other things that could be present as odor signatures of RDX. And that's the kind of thing that we've been doing in our laboratory, okay? We've been going into the instrument, to the ETD, and calibrating it and putting in compounds so that when they're present, when they're detected, it comes up with an alert. Okay, so that's what's been different.
Question for you?
Yeah.
Are there other products in the market that will have these plasticizers that won't be an explosive, though, that you may get a false reading?
Yes, there are.
Is there a fill, or how do you?
Well, that's the thing. So there is going to be a trade-off, and looking for compounds that you know are indicative of an explosive, but you know that they could be present in other things. So what you might do is look for combinations of things. So you might look for combinations of compounds that reduce that false positive rate. Yeah, the false positive rate is important. The false negative rate is more important. You don't want to let any explosives go through, right? It's just a matter of how much false positive can you tolerate.
So is your vision for this instrument to be a tool for prioritizing selective screening as opposed to more of a firm analysis of what you have?
All of these field detection tools are going to be presumptive screening tools. Ultimately, to confirm the presence of an explosive, that needs to go to a crime lab. So under controlled conditions, so you want to screen the guy before he gets on the airplane. You want to screen the guy who's in the car before he drives through the checkpoint. And then if you got a positive, then you continue the investigation. Okay, do a further search. You can even collect whatever items you want and then send those to the lab.
I agree. I've just seen that some decision makers make some decision. If you have an Odor and it says presumptively that this is window washing fluid, then they're satisfied to stop with any further action. And I'm just curious to see what your goal is if you're going to get to the level where people may make some decisions based on what they see, or you're just going to say there may or may not be energetic material present, and this requires a more complete investigation or separation.
That is really a policy kind of a decision. That's going to be what is the situation, what are the requirements for that particular situation, if it's a checkpoint screening, and then it's going to be by the agency is going to make that kind of determination. How do you progress when you have an alert with an instrument? So that's going to be agency by agency. What we're trying to do is provide an additional tool into your tool chest for detecting explosives. Will you get some false positives? Our indication is that is going to be very low. So, but we need to get this out in the field and continue to do this research. Okay, anything else? So here's what we want to do. is we want to mimic what the canines do. The canines cannot detect RDX. It's not available for detection. It's just simply not high enough vapor pressure. Okay? So the canines are trained on RDX, but it's the compounds that come off that are associated with the presence of RDX, C4 for example, that are being detected by the canines. And we found that what they alert to is 2-ethyl-1-hexanol for C4. So the dog thinks it's C4, but it's really 2-ethyl-1-hexanol. That's what we're trying to do with these instruments now, is we're trying to capture as much of the volatiles that are there as possible and then introduce them into the instrument so they can be detected. And that's exactly what the dogs do. And nobody, I mean, we know that the false positives for canines are improving as the training improves, okay? So if you have a dog alerting falsely, then you make adjustments to the training for that canine. In the airport, what we're trying to do is we'll alert mainly to the particles. So the canines are alerting to the vapors. In the airports, we're alerting to the particles. And in the laboratory, we're going to do both. We have the flexibility, we have the time, we have the controlled setting for be able to detect everything, okay? And that's a confirmatory test. In the field, we're doing really presumptive identifications, and then it's up to your policy to stop, evaluate further, and that's what you want to do. And that's exactly what the dogs do. So I'm not going to get into the chemistry of drugs, and you're probably all familiar with the different schedules of controlled substances and how they are placed into those schedules, and that's based on the potential for abuse and the accepted medical use. If a compound does not have any medical use and a high potential for abuse, they're placed in the highest category, which is Schedule 1, and examples of that are heroin, LSD, MDMA, and so forth. If there is a compound that has some medical use but still high potential for abuse, like cocaine, they're in the Schedule 2 and so forth. So we use a similar principle here. We want to be able to detect the presence of a hidden cocaine, for example, in a large container, and canines are the best detectors right now. We can also train canines to detect other things. MDMA does not have a very high vapor pressure, but we're going to train the canines to detect the compounds that are associated with the presence of MDMA, ecstasy. And Piperonal has been identified as a key volatile organic compound. It has a very high vapor pressure. It's available to be detected. So if the dogs can do it, we can design and experiment an instrumental system to do the same thing. So here are some volatiles from cocaine and marijuana. Whenever you see that you have no number here, whenever you see there's no number here, that means the vapor pressure is very low. That means the compound does not evaporate very easily. So we're going to target compounds that have a fairly high vapor pressure. So methylbenzoate. Here's methylbenzoate. It is a product of the composition of cocaine along with benzoic acid. So benzoic acid and methanol decompose out of cocaine when it's in acidic form, and you have any presence of water. They'll break apart and then they'll recombine to make methylbenzoate. Methylbenzoate is present pretty much in all cocaine as it decomposes over time. And that's what the dogs are alert to. We know that now. That's been published and confirmed. And you can train a dog on methylbenzoate and have the dog alert to cocaine. And we do the same thing with marijuana. So we're looking for these volatiles that are present in the plant that may be present in other plants, yes, but the combination of these compounds are going to alert to the possibility of having marijuana there, and then you can stop and do a search. Now, vapor pressure. This is the example I love to give. I have a 10-year-old son, and if I'm watching TV with my wife, watching a movie, and I make popcorn in the kitchen, he could be in his room, you know, several feet away. He can smell the popcorn. He comes out of the room. Where's the popcorn? He can smell it. Popcorn itself has very low vapor pressure. The popcorn kernel does not evaporate. However, there are compounds that come off of the popcorn that are very characteristic of the smell of popcorn, and we can smell them. What are they? Here's some of them. So that's what we do as a biological detector. Canines have a better ability to do this in that they can flush out their noses very quickly, and they can be trained, and the sensitivity of the canine olfaction is better than the human olfaction. So if you are looking for currency, if you're looking for cocaine on currency, many years ago we would look for cocaine on currency, then impound the currency because, well, this is illicit. It's been retrieved with illicit means. And now what we find is that much of the currency in circulation in the U.S. and in Europe is contaminated with cocaine, for example. Okay, so the drug gets adsorbed onto the bills, and then when the bills go to the bank and they're put into the machines, well, there's all this cross-contamination that happens. So a lot of the currency may have small particles of cocaine. What's interesting is that we still can impound currency that we believe has been, ill-gotten and associated with drug trafficking. And they do that by collecting the odors. Here's the dog. Here's the dog thinking it's got cocaine, but really they are alerting to methylbenzoate. Out of crack cocaine or powder cocaine, we have the odor emanates in the form of methylbenzoate. The dog is smelling the methylbenzoate. They think it's cocaine, but it's really methylbenzoate. Now, you may have 10 nanograms of cocaine on the surface. The dog's not going to smell that, not enough, okay? But if you have a lot of cocaine or a lot of recently contaminated bills with cocaine, then that quantity of odor will elicit an alert from the dogs. So this is kind of something we've learned over time, and my colleague at FIU, Ken Furton, has done a lot of research in this area, and there's actual case law now with the use of canines for being able to impound money that has been thought to be, illicit from illicit means. So we've also done a lot of work, and this is a project that was done in collaboration between our group at FIU and actually Patty Diaz and Michael Macias, who's now at the FBI laboratory, looked at the permeation rate of some devices. So we can put some illicit substances into a plastic bag and allow them to permeate out of the plastic bag. As we change the permeation rate, we can see that the canine response improves as we increase the permeation rate. So the more that is available, the more likely it is for the canine to alert. So this is what we call a dose response. The lower the dose, the lower the response. So as we go from 10 nanograms per second dose to the dog, we're only getting about 25% response. As we increase the dose, then we have a much better response. So this is a typical curve for a dose response between what you offer the dog and the response you get back. So Ken Furton's group has been working for years, as long as I've known him, maybe 15 years, on improving canine olfaction and canine alerts for both drugs, fire debris, explosives, etc. We do the same thing with instruments. We have the same kind of dose down here in the bottom, the amount in terms of nanograms that we expose to the instrument, and the intensity. So this is a typical calibration curve of an instrument. So you might have this as a background. This is a false positive right here. You're not exposing the instrument to any explosive, and you're still getting some signal, but we know to let the instrument know this is still a negative. So above some threshold, we'll say it's a positive, okay? And then the instrument will register an alert. You'll get the red screen, and you'll say there's something here. In the case of MDMA, the instrument is not normally detecting Piperonal, but we have put that in as one of the compounds for the instrument to detect. So now we can collect the odor signatures of MDMA. So we'll sample the air above a can that has some MDMA, 1 or 2 tablets, for a very short period of time, collect the Piperonal odor, place that sample into the instrument, and get an alert. So we've published a lot of this work. It's pretty exciting stuff. So Adam was talking about presumptive and confirmatory test. Field testing is usually presumptive. We're doing screening. Field detection can lead to further investigation and probable cause. In parentheses here, I am not an attorney. This is not a legal opinion. However, you may stop based on the policies in your agency and conduct further investigation, depending on what's going on. Confirmatory tests are usually conducted in an accredited laboratory under very controlled conditions by qualified scientists using standardized and validated methods, and they're the ones that usually go and testify along with you that made the first screening test as to the identity of the compound of interest. So how do I conclude this presentation? Well, there's a number of existing technologies out there for detecting drugs and explosives. Trace detectors target contamination from contact. So we're going to look for particles. If you have a bomb maker and they're not, extremely careful, they're going to get particles on the outside of the packaging, and that's where we're going to swab. And you'll see we have some particles that you can't see them. They're not visible, but we can detect them. Small amounts, nanograms, 10 to the minus 9 gram, a billionth of a gram can easily be detected with these trace detectors. So we're going to target particles for the presence of the RDX, and we're going to target vapors, the presence of the odor signatures that are associated with the presence of RDX, which is not going to be RDX. We can use biological detectors, and we can use instrumental detectors. And we feel that this new innovation, what we call the planar SPEMI, and you'll hear about it a little bit later, can be used for vapor detection when currently canines are the state of the art. And Brian has said, well, we're looking to see what these instrumental detectors can offer in comparison to the canines. Well, that's because instrumental detectors offer some advantages. They can operate 24/7. They're a lot less expensive once you get them up and running. And they don't require a handler. So there are some advantages of instrumental detectors over canines, even though we realize now as a community that canines are currently the state of the art. And remember, always the confirmation is done in the laboratory. Confirmation, you've got to collect that evidence and submit it to an accredited laboratory for confirmation. In your USB are all these lectures. So you can go back and look at all these slides again, and I'd like to point to some of the cited references here, some by our group and some by others, So if any of you are interested to go back and take a look at some of this original, peer-reviewed literature, we have some there. Any questions? No? Okay.